You can see in this second video, video 4.2, that I have the nucleus stated on the file, and there's this illustration of the nucleus. The nuclear envelope is what encloses the nucleus. It's a double membrane that surrounds the nucleus, keeps the contents of the nucleus separate from the rest of the cell. Little openings throughout the surface of the nuclear envelope are called nuclear pores. That allows things that are made in the nucleus to move outside the nucleus where they can be used in the cell, primarily RNA. You also notice that there's the nucleolus. The nucleolus stains darker than the rest of the nucleus. It contains RNA and protein. The watery portion surrounding the, well, actually inside the nucleus is called the nucleoplasm. It's analogous to the cytoplasm of the cell. There's a grainy looking material. It almost looks like there are little strands in here. The grainy material is actually the DNA and protein. It's called chromatin when it's not condensed into chromosomes. During mitosis, this chromatin condenses. It comes together into what we call chromosomes that you can see through a microscope. Otherwise, this just looks like a grainy material that we call chromatin. Same thing though. Inside the cell and outside the nucleus, we have what's called the cytoplasm. And the cytoplasm is everything between the plasma membrane and the nucleus. It does not include the nucleus. Suspended or floating around in that cytoplasm would be organelles, membrane-bound compartments that have specialized functions. A set of organelles that I want to talk about first belong to what's called the endomembrane system. And they're called the endomembrane system because they're very membranous and oftentimes connected with each other in where they begin and what they do. You can look up this image for yourself and look at the different organelles that are listed in the endomembrane system. The first one, and I should show here the endomembrane flow image. From the nucleus, we're going to have what's called endoplasmic reticulum, rough ER, that will be attached to it. It produces proteins. Those can be put into little membrane sacs, membrane-bound sacs called vesicles. Those can go into the Golgi apparatus. They're processed even further, and then eventually can come out as lysosomes, peroxisomes, or just some little vesicle that moves to the edge of the cell, joins with the plasma membrane, and the contents can be expelled. The first organelle would be the endoplasmic reticulum. There are two types. There's smooth and rough ER. The smooth endoplasmic reticulum, or smooth ER, as it's oftentimes referred to as, lacks ribosomes. The rough ER has ribosomes on its surface. That's why it's called rough, because to look at it, it looks rough. So the rough ER has these little dots on, and in an electron micrograph, they appear as little black dots, kind of like grains of sand or a little gravel. And the smooth endoplasmic reticulum lacks these. Because ribosomes are present on the rough ER, ribosomes are responsible in helping for protein synthesis. So whenever you see ribosomes, it should trigger something in your mind to remember proteins are associated with those. The smooth endoplasmic reticulum is responsible for lipid synthesis and for breakdown of toxins. And one of the things that leads scientists to understand that smooth ER is responsible for detoxifying the cells is because the cells of the liver have a lot of smooth ER. And one of the purposes of the liver is to detox the body. If you take in some kind of a toxin, then it's the liver primarily that breaks down that toxin. I tell my face-to-face students, if you give acetaminophen, Tylenol, to a cat, it will kill a cat because they don't have the enzymes produced by the liver to break down the acetaminophen. You give acetaminophen to a person, it's usually not a problem as long as they don't overdose, and as long as the person doesn't have high drug use, including alcohol. alcohol, other drug use can limit the function of the liver, and that's one of the reasons why doctors or nurses will take a history of a patient when they're admitted to a hospital, and they'll ask them about drug use, because that affects liver function, that affects what drugs they give them, and how many drugs, how much of a drug they can give them. The Golgi apparatus is sometimes associated with the smooth ER, sometimes there's some distance between it and the smooth ER, but it takes in materials from the endoplasmic reticulum and further processes them. And here you can see there's the cis face and the trans face. The cis face is where vesicles or materials are coming in, chemicals are coming in, they're absorbed, they're put through this system, this Golgi apparatus, where they're further processed, chemical reactions occur, they're put out into a little vesicle that's secreted from the trans face of the Golgi apparatus, and then these either stay in the cell or they can move to the plasma membrane and be expelled. The vesicle is just a small membrane-bound sac, and we tend to think of these in animals. They can be in plants also, but we tend to think of them in animals associated with the endomembrane system. Lysosomes are vesicles that will have hydrolytic enzymes in them to break down materials. They may break down waste that are in the cells. They may break down bacteria that have been taken in by white blood cells, but very important in digesting or hydrolyzing materials. It's been taught in the past that when a cell dies, the lysosomes break open and those enzymes auto-digest the cell. This is part of the explanation for hanging meat and allowing it to hang for a number of days. It actually will help the meat to be more tender. And part of the explanation is these lysosomes are being released. They break down the protein, they break down the cells, and the meat is more tender. We're in a big deer hunting area in southern Illinois. A lot of people like to hunt deer. If you're able to hang the deer for several days, it improves the texture and even the taste of the meat. A lot of people like to do that. Here is an electron micrograph of a lysosome, and there's some kind of an organelle fragment here and another organelle fragment here that the lysosome is breaking down. Other vacuoles will have enzymes similar to catalase that will break down hydrogen peroxide, and those are called peroxisomes. And in this electron micrograph, you see this crystal structure. It's got what appears to be 6 sides. It's a hexagram. Hex means 6. It's got these 6 sides to it, and this is typical for peroxisomes. If you look up more information on peroxisomes, they'll show a crystalline-like substance that is found inside the peroxisome. An interesting pattern. It doesn't show up here very well because I'm zoomed out, but if you were to go to this image on the internet and look at it and enlarge it, you'd see an interesting little pattern to it. Many eukaryotic cells have peroxisomes, and that's to break down hydrogen peroxide that's produced as part of the metabolic process. Vacuole, that's a large storage compartment. It can store waste, it can store nutrients, it can store acids, it can store a number of things, stains from plant cells, and can take up a vast majority of the cell and push other organelles off to the side. Here's a vacuole in a plant cell, and you can see it's taking up a good amount of the volume and pushing the chloroplasts off to the side. You can see these chloroplasts. We'll talk about those a little bit later. Some organelles that are concerned with energy transformation, and I'm through with the endomembrane system. The chloroplast is very important in transforming light energy into chemical energy, and there are several parts to the chloroplast that we want to be familiar with. The stroma is the watery portion inside the chloroplast, and it should be noted the chloroplast has a double membrane around it. There are structures inside the chloroplast called grana, granum singular, and these are stacks of thylakoids. These thylakoids are disc or coin-shaped structures, very important in photosynthesis in what are called the light reactions. With the thylakoids, a stack of these is called a granum, and these are found floating around just in the stroma. Here's an electron micrograph of a chloroplast, and you can see the grana. They're a little bit darker, seem to be connected with these strands, part of the cytoskeleton. And let me see. Okay. This is from through a light microscope. The chloroplasts are green because they're reflecting green light, and that's because of the chlorophyll in the chloroplasts. And if we had some plant cells, especially algae that's found in the water from a pond, maybe a local pond, we might even be able to see some cyclosis or cell streaming. You can see these chloroplasts right here are in a line. Sometimes they'll start moving around the periphery of the cell. It looks like there are a bunch over here that are in a line. Don't see it very often. If I had about 20 to 30 students, I might see it in two slides perhaps. Another organelle that's very important in energy transformation would be the mitochondrion. Mitochondria would actually be plural. And the mitochondrion has a double membrane. The inner membrane has foldings, or has folds to it, and each fold is called a crista. Cristae would be plural. And those foldings of the crista, or actually of the inner membrane, called cristae, greatly increase the surface area over which chemical reactions can occur. The watery portion inside that inner membrane is called the matrix. And these areas become important later on in the semester when we talk about cellular respiration, and for the chloroplast, and the parts of the chloroplast when we talk about photosynthesis. The mitochondrion is called the power plant of the cell because it provides the energy, most of the energy for the cell. Mitochondrial DNA is designated with a lowercase m DNA, and there are different types of DNA, and for the DNA that's found in the mitochondrion, it's called mitochondrial DNA. That's important. It's been studied for a number of years, going back into the 1980s. Research was done around the world where researchers went into villages and cities around the world, including Africa and Asia, and noted some people lived and died within a 20-mile radius of where they were born. And the mitochondrial DNA was compared amongst different people and different groups around the earth, and scientists determined that everybody came from one female, and they actually called her Mitochondrial Eve, an interesting expression that was applied to her. And they claim that she lived in Africa. We don't know all that, but that's what's been surmised decades ago, back in the 80s. And I think it was the name of the journal is on the edge of my memory. I can't think of the name of it, but I had students go down to the library and read it. I had it on reserve for a number of years, but it's no longer there. If I can think of it, I'll mention it later. Mitochondrial DNA becomes important because it is used to link people to crimes when the DNA is aged. There was a crime that happened in the late 80s in our area. It involved a young girl down around Kell. She was molested and murdered. And because so much time had elapsed from the time the crime had taken place until the appeal, the convicted person was granted another trial. It had been decades, and so they couldn't use regular DNA. They used mitochondrial DNA and tried to match his dog to hairs that were found on the young girl. It is also important to note that mitochondrial DNA is only found in mitochondria, and all the mitochondria in a person's body arose from mitochondria from the ovum, meaning whether you're male or female, all of your mitochondria come from your mother. So that makes for interesting studies. I'll continue on the next video.